Hi, welcome to Inside the Classroom. I'm Courtney Simpson and this is my AP Biology class, and they are in the second part of a lab called Mission Impossible. And basically what they have is they have two tubes that have unknown pieces of DNA, and they treated the tubes with different restriction enzymes, and so now they're going to start the second part of the experiment, which is gel electrophoresis. And they're going to, at the end of the block, they're going to look at their gels just to try to figure out what pieces of DNA they had in each tube. Welcome inside the classroom for your tubes. And there's a couple of things you need to do. We did not have the ladder in the last class, the DNA ladder, so you need to add 18 microliters of the distilled water to the ladder tube. And then to every single tube, so 5 total tubes, you're going to add 4 microliters of the loading dye, and the loading dye will allow you to see where the DNA might be moving on the gel, because you can't see the DNA because it's invisible. And after you add the loading dye, you're then going to put all 5 tubes in the centrifuge, pulse it for just a second, and then you will load 15 microliters of each sample into the well. And I gave you on the lab 2 choices as to how you want to load your gel, so you can refer to that in the lab. And then we'll set up the gel electrophoresis, plug it in, and then in approximately 35 minutes we will look at the results of the gel on the UV light. Make sure you change pipette tips every single time. Okay?
Yeah.
Okay.
Ready?
Go to it. Sabine, you're over there with the. Okay.
We don't have them.
Yes.
Okay, right?
You wrote it on the side.
I mean, it's just not good.
And I will put the gels into your little chamber.
Okay.
I did all of this by myself yesterday.
Two over here.
Do you have your partner today? No. I saw her yesterday.
And we need 4 for
She's gonna be here today, but she ditched me yesterday.
Okay.
Do we need gloves?
Let's do this. I'm ready to go. Full lab partner.
What are we doing?
And you don't need goggles either.
What's on the board?
Okay. Is that our microphone?
Ladder. Add 8 adding the micropipettes to the right amount of water that we want to add to the DNA ladder.
Yeah.
Yeah, this is like reality knocks hard.
Oh, do you want to do this?
Yeah, sure.
Yeah.
Okay, so you did that.
Yeah, it's set to 18, so just push down the first stop.
Yeah, I know it takes a while.
Did you notice, Macaulay, that we're adding 18 microliters of GH2O to our DNA ladder?
Which is distilled water, by the way.
Yeah, GH2O is distilled water.
Yeah. I was reading the book.
Nice. I just wanted to make sure.
Someone messed up. It's supposed to say LAD,
Okay.
but it just says LB.
It was also here, so I was like, cool.
So the ladder is
Oh,
DNA.
did you push it in the second stop?
What's going on with her?
That's okay. No, it's okay. Just.
I think it's 80, yeah.
So we're making a DNA ladder, which is going to be what we compare where the other enzymes go on the gel electrophoresis to figure out the number of base pairs for each plasmid fragment.
You need to add 2 tubes. I summarized on that.
Never seen that. I was in biotech.
Okay.
Keep it like that, and then I'll just go.
You're going to put change shifts.
So, and you're gonna go second stop. Yeah, just like put the little tip on the side too. That's how I like to do it. Second stop. Then second stop. And then keep it down, lift it. Yeah, that's. I mean, we got like.
It takes forever because you're
So here, I'll give you
change the tip.
Okay, let's just.
sorry.
You just hit the
Oh, I thought it was this.
And here.
All you do is
I stab it on.
I like going to 18 because then the stop is like not
So we need to
existent.
put
Still 4.
18 microliters
Yeah,
back
4.
into LAD. But since you're setting it to 4 anyway, just do the 4.
Okay, and then you're gonna put
We're gonna do
LT
distilled water now.
in all 5 of these.
Okay, just 4.
Yeah, but you're gonna change chips
Every
time.
single time.
Add water.
Okay, so we'll go back to Jacob.
Yeah, it's a camera. Yeah.
Okay, so I'm gonna get you guys in the gels.
And
Okay, and make sure you push that inside.
Nothing came out. Okay,
You know,
okay.
thank
How
you,
about,
just gave us a clean start.
Remember when you're loading the wells, you only go to the first stop.
okay, yeah, there's nothing in there.
The challenging part about this project is if you're doing it alone and you have to close all of the different tubes by yourself, especially if you're trying to get loading dye and then change your tips mid-process because you have to change your tip every time that you go into a new tube. And if you're doing it one-handed, then you kind of got to help yourself.
Wait, but Miss Simpson, can we borrow your camera this weekend?
So today we are going to be doing a gel electrophoresis experiment, which is where we observe DNA and how it moves on a gel, which is made out of agarose, a seaweed powder. And we're hoping to see which restriction enzymes cut a given plasmid into different pieces, and then identify the mysterious plasmid.
Hard doing it one person. It's like some of them are like seriously stuck too. Like yeah.
What happened?
Wait, you need your body.
Clean it out really quick.
This one?
Yeah, I'll just do it really quick. So I have to get to 15.
You're supposed to add it to the ladder. 4 microliters of dye goes into every single.
Oh, okay, good.
Oh, okay, that's why I was like, why is
Yeah, I was
this
so
working? 'Cause it
confused.
should've. Okay, so then we can put it back on top.
Yeah. No, I know.
That one will go right there. Wait, so now the names are on the stuff, but it's not really.
Yeah.
On the first go.
No, I can do it.
Just put 18 of the
Oh,
silver.
18, that's why.
Yeah. How are you doing?
18 of distilled
Yes.
water into
Okay.
DNA.
So you put glue in all
Because then that's I'll have 5 and then that's what.
Yeah.
With a thing on it.
Okay.
18 into
Did you do them?
Yeah, I just have to put dye in the last one.
It's a little tedious just because we have to make sure that everything is exact, and a lot can go wrong because there's like the slightest mistake. So, because we only have like so much of each reagent, so we can't really afford to mess anything up.
Well, that's
So in that case, it can be a little bit nerve-wracking.
Yeah, we're using less than 1 milliliter of fluid in the solution, so it's really frustrating.
Yeah,
Oh,
So now I have to wait.
2 out of 5.
Yeah.
Goody.
And then.
So this system right here, this is an old electrophoresis system. It's about 10 years old. And you basically have to hook it up to a battery, which then powers the electrical current to separate the DNA fragments. Whereas I just recently wrote a grant and we got these over here. And it's kind of like an all-in-one system, and these are great because you use a lot less agarose and you use a lot less buffer, so less reagents, more economical. And the cool thing about these is it's an all-in-one system, so you can turn it on and you can actually, you won't see anything right now, but this light exposes the DNA to UV light, so it will actually glow and you don't have to, it's like an all-in-one system, whereas those old ones, we have to take the gel out and put it on a UV light, whereas here you can actually see everything working in real time. 50 will fit. You might want to hold when you have your phone to shine like a light.
She's filling up the tubes with the certain enzymes
and mixed with DNA. And what will happen is when the machine gets turned on, they'll move up the ladder, and we'll be able to tell which DNA sample is which because it'll be cut at different lengths by the enzyme.
And really most important thing is
We should load it like a picture.
Sorry.
yes. Right? Did you not add the dye to it?
So do
You
like.
have to add loading dye to the ladder. Did you suck up that little tiny drop? Say that again?
So do the ladder the second.
The little ladder that's in there.
Yeah.
The second one in. Tiny little drop you sucked up there. Okay, there's no way you can suck up 15.
I guess touch the ladder, but I don't think it broke.
The second one is gonna like blow all the water.
Do you like to look at it like this, or what you're gonna do?
Well,
Drop loading dye and add it to here. Yeah, did you mix it?
It's going to be 15.
How it is on the chart.
Yeah, and maybe
Do I need to
skip
do
a
some
basis. No, you don't have to,
You
but.
add some water. Get rid of this. Okay. You like to add a
I add 18.
So you squirted it back in here. Okay, and now you need to add loading dye.
Okay, so yeah, so it's just the dot.
Yes.
Yeah.
Okay, yeah.
I added something. So the best way to mess it is
No, this one right next to that one.
Yeah.
So you can either put them all like in a line next to each other, or we can have like a one space.
Alright.
So we're right next to it and every other.
Yeah.
15. So with these, when you load it, or when you put it into the gel, you only go to the first stop instead of the second stop for the tubes.
Nice.
If you go to the second, like,
So what we're doing is we're loading the DNA samples into the gel, and then what we're going to do is we're going to hook it up to an electric source so that it'll pull the DNA away, and then the smaller fragments of DNA are going to move farther toward the other end, and then the longer DNA fragments are going to move closer to where the wells are. So that's going to be how we determine which tube is which type of DNA. You put the top on.
Yeah. Wait, we put the top one now?
Yep. You're gonna slide it in.
So I think there's probably the nature of the pipette tips.
And then we just have to wait,
The second I touch the water, just
because there's only one, like,
That'll be
battery,
fine, but
I
just-
think, for one power source. So you have to wait.
You don't get any better. No, here, wait, try to do this.
To discard.
The second
it's
one
just gonna back.
Yeah, because the second I just did that, it just, it's.
I put it into it.
So now the first one we're gonna do is a
Yes.
enzyme.
And it's the same amount. Never
Yep.
go to the second stop.
It's fine if it comes out as such a No, it just hits and it just comes out. Right, and it falls down.
Miss Simpson, do we look like new gel electrophoresis?
I'm Luke Fournier, I'm a senior, and I think I enjoy most just working on a really small scale. I think it's awesome. That's it.
Nice job.
So right now we're going to be waiting for the DNA to separate through the electrical currents, and the plasmid that we cut with restriction enzymes will be split into either 2 or 3 parts depending on where along the fragment of DNA it was cut. So after this is done running, we'll see multiple fragments of DNA in each well. There'll be a bunch here and then 2 or 3 in the other ones.
So this was my estimation of where when the plasmid would be cut, like where the bands would move to. So like we use SNAP and Gluc enzymes, or like to cut them, but so there should be 2 in this because it's the enzyme, and then 1 in the control, and then 2 in the enzyme and 1 in the control again. But then depending on what enzyme we use to cut them, they'll be farther, like spaced apart or not. So like if they're smaller, they'll be down here more, or if they're like larger, they'll be closer to the wells.
Okay, so now what they're going to do is the gel electrophoresis has been running for about 35 minutes, and they're going to take the gels out of the chambers, that's the beep actually, and they're going to expose them to UV light, because we stained the DNA with a, something called gel green, and when it's exposed to UV light it glows. So that's how they're going to be able to see where their DNA is on the gel. Alright.
I'll send you a link where I have it. Yeah. Luke. I told him about it. He's like, what's it called? I was like, I'm not gonna tell you.
Whoa, that is okay. I was supposed to take that off.
That's real UV light.
Yeah, it is.
Do not let your gel slide off. We had one accident in the last class.
Wait, Miss Simpson, we like can't see it.
Is it too foggy?
Yeah.
Okay, so what we're gonna do is you're gonna just shut it off for a second. Just stop.
Oh yeah, we turned
Yeah, I
it
made
off.
that mistake.
And
Should I
then
take
wipe
the
it out.
lid
That's very good.
Take the lid off, wipe it off with a piece of paper towel, and then put it back on and turn the light on.
It looks.
Oh yeah.
So right now we're looking at the completed gel electrophoresis experiment, and the DNA has been basically exposed to an electric current and has separated into different pieces. So the plasmids are now, that have been cut with restriction enzymes, are going to be in different places along the gel based on how big they are.
Yeah. Oh.
We bring it over?
Yeah, because it only goes like that.
Yeah. Wait, did you want us to cut it first?
I thought we messed up because all ours was the same.
It's okay.
I should have grabbed some of this one. Oh wait, Jack, grab that.
So now we're going to analyze the results of our gel electrophoresis experiment. As you can see here, all the way on the left is the DNA ladder, which we'll use to measure the different bands. And then here we have the control, the enzyme, another control, and then another enzyme cut plasmid. And you can see they're all spaced out at different places, which is how big they were based on which restriction enzymes we used.
Definitely glue. Next snap is that.
And there's two ones pretty close to each other.
Yeah, and it's like 18 and
What enzyme did you use?
I used a different one. I used
Oh, this is yours. Which enzyme did you use?
Oh, XMA1
And BAM.
and BAM.
And BAM.
Oh, we
Okay.
only used X.
So what did you expect to see?
We expect to see the ladder and then A enzyme, so 2.
Two.
A control 1. S enzyme 3. S control 1.
Okay, so then, so can you tell which one is P snap and glue based on?
It would be snap.
Okay, so make sure you take a picture of that.
So under the light, if this first one is the ladder, so that tells us that's what we compare the sizes to. And so this first one was cut with 2 enzymes because it's a circular piece of DNA. So if it was cut twice, that would create 2 fragments, which it did. And this control had no enzymes in it, so that's why it's 1 fragment. And then this one was cut 3 times. That's why it created 3 fragments of the DNA, and this one was also not cut, and that's why I only had 1 fragment.
Because our lines were really clearly cut, we can now see which enzyme is which. It's been a successful lab. Thank you for joining us today inside the classroom, and hopefully you see here at NASA that we work with high-tech equipment and we're doing high-tech labs here in AP Biology.